Continuing where we left off in our previous video, we will now look at the virulence factors that allow bacteria to survive inside the host and evade the attempts by the immune system to get rid of it, inactivate it, kill it. So bacteria can do several different things that allow them to evade capture by the phagocytes. So just as a refresher, the phagocytes are those that are able to ingest and destroy bacteria. One way that bacteria can evade these phagocytes is by producing capsules. Capsules help with adhesion, as we looked at, but they also prevent the ingestion by phagocytes. The immune cells can't stick to that capsule, and if they can't stick to it, then they can't ingest it. The capsule also makes the bacterial cell larger, which also inhibits the action of the phagocytes. One example of a capsule-producing bacterium is Streptococcus pneumoniae, which causes pneumonia and several other diseases. The strains of S. pneumoniae that have the capsule are much more virulent and likely to cause disease than those that do not have the capsule. Other pathogens can produce proteases, which are enzymes that break down proteins. The immune system produces antibodies that bind to these different components of bacterial cells, and that initiates the immune response. These proteases essentially break down the antibody molecules so they're not able to do their job. Fimbriae of some Streptococcus bacteria contain a special protein called M protein, and it actually makes the bacterium look different or appear different to the immune system and prevents phagocytosis. Mycolic acid, which we know is produced by our Mycobacterium species, will allow the bacterium to actually live or survive inside of the phagocyte. So it doesn't block the action of phagocytosis, but once the bacterium is inside the phagocyte, it prevents its destruction. So it's essentially just growing and reproducing inside that phagocyte. Some bacteria will produce virulence factors that actually exploit our own molecules and try to basically outwit the body's defenses that way. A lot of strains of Staphylococcus aureus produce coagulase. Coagulase breaks up or stops the process of coagulation, which is the natural mechanism of blood clotting. In the circumstance here with Staphylococcus aureus, the bacteria essentially triggers the formation of a blood clot, which encapsulates the bacteria in fibrin and essentially allows it to hide from the immune system. So they can basically outsmart or outwit, our body's own mechanisms. Another way that pathogens can protect themselves from the immune system is called antigenic variation. Antigenic variation is when the pathogen alters the proteins on its surface so that the immune system no longer recognizes it. This would be essentially changing its coat. Okay, so if you have a bacterium that wore a blue coat, okay, and the immune system recognized that blue coat, then it would start to produce antibodies against the blue coat, and when it encountered those blue-coated bacteria, then it would attack and launch a response. Well, the bacterium then can take off that blue coat and put on a purple coat. And even though it's the same bacterium, the immune system doesn't recognize it anymore because it's looking for a blue coat. There are no more blue coats. They're all purple coats. So the immune system then has to learn that this bacterium now has a purple coat. So it will start to produce these antibodies against the purple coat and start to get a foothold on the infection. Well, then the bacterium can change to a red coat, and so on and so on and so on. So every time the bacterium changes its surface proteins, the immune system essentially has to learn it all over again. So it avoids the immune system response because the immune system doesn't recognize it anymore. Now, switching gears a little bit, looking at some of the virulence factors of viral pathogens. Some of the mechanisms and properties that viruses use are similar to bacteria. For instance, viruses also have adhesins. Remember that in our viral replication cycle, we looked at the first step was to adhere to the host cell. And there are different types of adhesins that viruses have that allow them to attach to the host cell. For example, we have hemagglutinin, or the H that we refer to when we talk about influenza viruses. Hemagglutinin allows the influenza virus to bind to a specific acid on the membrane of host cells. And this is referred to as a spike protein. You've probably heard about these in the news a lot because coronaviruses also use spike proteins to attach to their host cells. And in fact, a lot of viruses do. So viruses also need adhesins. If they can't attach, if they can't adhere to the host cell, then they can't cause disease. They can't infect our cells. Viruses can also exhibit antigenic variation, and it's pretty specific in terms of what viruses and what types of viruses can do this. We see this in enveloped viruses, and again, one of the best studied antigenic variations occurs within the influenza viruses. Influenza viruses are able to undergo two different types of antigenic variation: antigenic drift and antigenic shift. So these two are illustrated for you on the screen here. Antigenic drift are slight changes in the spike proteins H and N. So this is typically when there's going to be some type of point mutation that occurs. It's going to be a very small mutation within the viral genome, and we're going to get just a very slight change. So here would be an example of antigenic drift. Notice the shape of our neuraminidase, the N, and then the hemagglutinin, the H. The neuraminidase is kind of oval, and the hemagglutinin is kind of like a pentagon shape there. And there's a little change in the DNA, and now the hemagglutinin looks kind of like a little flower. Okay, so that would be antigenic drift. It's just a very slight change. It's the same H, it's the same N, but the H just has a slightly different shape. Antigenic shift, on the other hand, is a major change in the spike proteins that occurs as a result of genes being basically reassorted. This typically happens when we get two different strains of the influenza virus that infect the same host. Okay, so we have virus A here with our oval-shaped N proteins and our pentagon-shaped H proteins. And we have another influenza virus that has also the same shape, but we've got them illustrated here in a different color. Okay, so they basically have different N and H proteins. They both infect the same host, and so they, inject their material into the host, and as the virus is replicating, it just jumbles all of these genes up together. So now we have an entirely different strain. We have the N, the neuraminidase from virus A, okay, our yellow ovals here, but the hemagglutinin, the H, is from virus B. We've got these blue pentagons here. So this is antigenic shift. We have a brand new strain of the virus. So when you hear on, the news or read on the CDC website about these flu viruses, you'll see them, H3N2 or H5N2 or H1N1 or whatever. And what those are referring to are the particular types of hemagglutinin and neuraminidase spike proteins. Every now and then, we'll get a brand new virus that comes out because of antigenic shift. And we don't just see it in humans. The flu virus can infect pigs, it can infect birds, and so that's why you hear about, swine flu or bird flu. So they can play a big role in antigenic shift. And this is why, we have new flu vaccines, because we may develop some immunity to this strain, or maybe we have immunity to this strain here. But when this new virus comes out, we don't have immunity to that one anymore because it looks, completely different. So antigenic drift and antigenic shift occurs differently in viruses than it does in bacteria.